Welcome back, students. This is Module 1.2.7 called Lipids. And last time we introduced you to biological macromolecules by talking about carbohydrates. Today we're going to further your understanding of biological macromolecules by studying lipids. Lipids have a variety of functions. They function in energy storage, they function in insulation and in padding for vital organs, especially your kidneys. They're important structurally in cell membranes, a critical function of lipids. Lipids are insoluble in water because lipids are nonpolar and water is polar. So if you go back to our general chemistry introduction and look at the attributes of water, it will help you to understand the old adage that oil and water don't mix. This is so true in chemistry that we have a technical term that describes this. That term is hydrophobic. Of course, if we look etymologically at the word roots that make up the word hydrophobic, hydro is water and phobic is fearing. And so this term, hydrophobic, actually describes the behavior of nonpolar molecules' behavior in water. And so oil will avoid water. Very, very important idea here. And that's one attribute that all lipids share that helps to group them. They are a rather diverse and varied group of molecules. We're going to look at three major groups of lipids. We're going to look at triglycerides, phospholipids, and steroids. Could put number 4 waxes, but we're not going to really look at them. Waxes are very unusual. Each one has pretty much its own kind of structure, and they have varied functions from beeswax to earwax. But that's all we're really going to discuss them. We're going to start by talking about triglycerides. Triglycerides are fats and oils, and they consist of a backbone of glycerol, and they have 3 fatty acid chains. And if you look at the name triglyceride, it means 3 and glycerol. So from this backbone of glycerol, they have these 3 fatty acid chains. One of the things that it's important for you to do in this class is we would like for you to be able to distinguish between fats and oils, because both are triglycerides, but what is the difference? In layman's terms, fats are typically solid at room temperature and oils are typically liquid at room temperature, but we want your understanding to go beyond that. You certainly should know that. But fats are solid at room temperature because the chains of fatty acids are saturated. And when we use the word saturated in this context, what that means is that each carbon in that chain has as many hydrogens as it can possibly hold. And these tend to be solid at room temperature, as I said already. And examples of that I didn't give you are things like lard and butter. Okay, and this is just definitional. We're going to look at diagrams in a moment, and this will help you to understand this concept. And I'll tell you why having a saturated chain tends to make molecules solid at room temperature. While oils, by contrast, the chains, the fatty acid chains, are unsaturated. At least one or more of the carbons that are in those fatty acid chains of oils have double bonds. And this means that they don't hold the maximum amount of hydrogen. They are not saturated, hence they are unsaturated. And this causes a molecule to be liquid at room temperature. And this has to do with the fact that double bonds change the bond angle of the tails. And the tails can't be packed as closely together in unsaturated fatty acid tails as they can in saturated. And remember the difference between a solid and a liquid, at least one of them, was how densely packed the molecules are. If you can't pack them as tightly, then you're more likely to be liquid. And if you can pack the molecules or the atoms more tightly, then you're more likely to be solid. And again, we'll show you an illustration of this. We'll come back to the other point on this slide. So this is actually, so you can put an image to this idea of a triglyceride. This shows the formation of a triglyceride by dehydration synthesis. You can see it loses 3 drops of water. 3 drops of water are liberated, and 3 bonds are formed, and so glycerol was the backbone, and we put 3 fatty acid tails on there. And so this is kind of the process that we're seeing. This actually shows a little prairie dog, and it shows a fat vacuole, and what is, of course, stored in a fat vacuole, and the way that mammals, including humans, store fat is as triglycerides. So this is an important thing to understand. Let's further explore the structure of triglycerides in this next illustration. Again, in the upper left here, where the cursor is moving, there's the glycerol, and here is one fatty acid. And fatty acids are all named, and we're not going to get into any of that nomenclature. But this shows a simple triglyceride, the glycerol with 3 fatty acid tails. And this one, by the angle here, how tightly packed they are, these are saturated. Now, in biochemistry, the shorthand is when you draw a zigzag line like this, each corner represents a carbon. Each carbon has a valence of 4, which is to say that it can make 4 bonds. So 4 atoms can attach to each carbon. And if you look down here at this picture, on the very bottom center here, I've shown you 3 carbons. 2 of the carbons have a single bond between them, these 2 on the right, and the center one and the one on the left are double bonded. Now, a double bond is something that we have not specifically described before, but a double bond, in a double bond, 2 atoms share 2 pairs of electrons. Remember, in a regular covalent bond, a single covalent bond, 2 atoms share 1 pair of electrons, but here they're sharing 2. And so we can see, of course, this counts as 2 bonds. So the carbon on the right, we can count 1, 2, 3, 4 bonds. The carbon in the center, 1, 2, 3, 4, because there are 2 double bonded there. Now we can see that this is not saturated because this double bond could in fact be broken and another hydrogen could be present on these 2 carbons in the center and on the left. Now, the thing about double bonds is it changes the bond angle. If you look at the diagram in the upper right here, where the cursor is, you can see I'm pointing at the double bond. This is labeled as kink, and all that means is that the bond angle is different, and so it's further apart. Therefore, it tends to be liquid at room temperature rather than solid. So the structure of a saturated triglyceride here, the structure of an unsaturated triglyceride here. So I hope that you could explain why different triglycerides are solid or liquid at room temperature, and also what saturated and unsaturated means. Let me say one last thing about this, the health consequences of eating excess fat. A doctor will typically recommend to you that you reduce all forms of fat. Saturated fats, because your bloodstream contains blood, which is water-based, it has a difficult time transporting especially saturated fats. And if you eat a diet high in saturated fats, you can get occlusions or blockages in your blood vessels, and this can lead to things like heart attacks if the blood vessel that's blocked is the one that supplies the blood to the heart muscle. So definitely you shouldn't eat too many saturated fats. Some of the unsaturated fats also can have an adverse effect on the cardiovascular health, the health of the heart and the blood vessels. But especially some of them have been linked with certain types of cancer, so you really do want to reduce your intake of both types. So let us return to this slide and look at the last bulleted point here. And this is an important fact for you to recognize, because one of the things that you may read before you go to classes in the morning is your cereal box. And on the cereal box it talks about the proportion of different types of biological macromolecules that are found in your foods. And one of the things that you should recognize is that fat has more caloric density, which is to say that for the same amount of weight, for 1 gram, fat has 9 calories, 9 kilocalories, and protein and carbohydrate both have about 4 kilocalories. Now this kilocalorie with the K and the capital C is the same as calories that you count on a diet. So 1 kilocalorie is 1 calorie as you count on a diet. So 1 gram of fat has 9 kilocalories or 9 calories, and 1 gram of carbohydrate or protein has 4 kilocalories. And so this explains why animals which are mobile, which walk around, preferably store their energy as lipids. It's because you get more storage for each unit of weight. It's more efficient. Okay, let's move on and talk about a second type of lipids. So we're going to leave triglycerides. We're going to discuss phospholipids. Phospholipids have a backbone with a hydrophilic phosphate head. Now, we haven't defined the word hydrophilic, but it means water-loving, and a hydrophobic tail that consists of 2 fatty acid chains. Here, instead of drawing the zigzag, we've just drawn a straight line. So the blue sphere here represents the phosphate head, and the 2 lines represent the fatty acid tails, and they're hydrophobic. And so we can see these put together below. And if you put a bunch of phospholipids into a watery environment, they will turn their hydrophobic tails inward, and they will form what we call a lipid bilayer. And that is what is illustrated here. The spheres on the outside are the hydrophilic heads, and the lines on the inside are 2 layers of hydrophobic tails. And phospholipids play a critical role in forming cell membranes. And we'll talk more about this when we talk about cells and cell transport. So we will return to this. But you should understand the basic idea, the basic structure of a phospholipid as I've described it here. Now, very quickly, we will describe a third type of lipid, a category of lipid, which are steroids. Steroids have important roles as messengers, and they also can have a structural role. Hormones are powerful chemical messengers, and some of the sex hormones, estrogen and testosterone, are both steroids. And you can see steroids if you look down here in this blue box in the bottom center of the slide. Steroids tend to have this 4-ring structure, kind of a distinctive structure, if you will. And as I said, they're important chemical messengers. It is the sex hormones, which are steroids, which provide clues, and I should say cues rather than clues, for secondary sexual characteristics to develop. And they have many other important functions as well. We can look at a second role. I mentioned just previously that some steroids can be structural. And one such structural steroid is cholesterol. And we've heard that cholesterol can cause a problem, and it can. It can also lead, because it is a lipid, and if you have excess cholesterol, it too can cause blockages in blood vessels and cause cardiovascular problems. And so sometimes I ask my students, if you could get rid of all of the cholesterol in your body, should you do it? And of course the answer is no, because you need cholesterol. No one has ever died from a shortage of cholesterol, because animal cells can make all a sufficient amount of cholesterol. We can see, if we look at this picture on the lower right here, this should look somewhat familiar. These spheres are the phosphate heads of phospholipids. This is a cell membrane, and it's showing cholesterol that's stuck inside there. And what cholesterol does, the more cholesterol there is, the further apart these fatty acid tails are, making the membrane more fluid. Now, this is important because in different temperatures, in colder temperatures, the membrane tends to become more solid, and so the amount of cholesterol is increased in a cooler environment. It's kind of an interesting phenomenon. And with that, we're going to conclude our discussion of lipids and conclude this module. We will continue with our last module on biochemistry a little later.